bundle up to form your muscle fibers. They're divided lengthwise into segments called sarcomeres, which contain two even tinier strands of protein, two different kinds of thin filaments made up mostly of two light and twisty actin strands, and thick filaments composed of thicker, lumpy-looking myosin strands. Each sarcomere is separated by what's known as a Z-line. Sarcomeres contracting, bringing those Z-lines. Namely, actin is blocked by a couple of protein bodyguards called tropomyosin and troponin, which keep getting in the way of nuclei, but some of them also have a lot of mitochondria, whose sole purpose in life is utilized, so it gets a special name, the sarcoplasmic reticulum. Its walls are loaded with calcium pumps, which use ATP to receptors on that muscle cell are ligand-gated sodium channels. So when the motor neuron releases our old friend acetylcholine into the synapse, the channels open up and create a rush of sodium into the cell as a graded potential, which has lots of tubes that run deep inside the cell, called T-tubules. When the action potential travels down one of those tubes, it eventually triggers the voltage-sensitive proteins that are linked to those calcium channels in the cell's sarcoplasmic reticulum. Bodyguard protein, the tropomyosin, away from the sites on the actin strands that the myosin really wants to get its paws on. It moves into an extended position, kind of like a stretched spring, still holding on to the ADP and the phosphate, and still storing the pulling a rope hand over fist. In the process, it shrinks the whole sarcomere, and be spent, that little head has no use for the ADP and the phosphate, so they unbind with it. That unbinding causes a small change in its shape, which lets a fresh ATP bind there in its place. That binding causes another shape change, but this time it causes the myosin to release from the actin ATP into ADP and a phosphate, which moves it into the armed position yet again, getting it ready for its next rendezvous. They start grabbing the calcium that's floating around, causing calcium to unbind from the troponin. When it unbinds, the resulting shape change puts the tropomyosin bodyguards back